Hello, and a very warm welcome to everyone joining us for this SelectScience Roundtable event titled How to Avoid the Costly Pitfalls of Assay Scale-Up. My name is Cameron Smith. I'll be moderating today's presentation, and I'm delighted to be joined by all of our guest speakers. Joining us today, we have Brian Cataneo, New Product Development Staff Engineer at TE-IBD Solutions, Michaela Hoffmeyer, Director of Regulatory Affairs at TE-IBD Solutions, Dr. Fraser Young, Senior Scientist and Lyophilization Specialist at Biofortuna, and of course, Dipankar Manna, Senior Director of Research and Development at LGC Biosearch Technologies. Throughout the session today, please feel free to submit any questions you may have. To do this, simply hover over the purple speech bubble icon on your screen and click to open the submission tool. There you can type your question and submit them for the Q&A sessions later on. So without further delay, I'm pleased to kick off today's presentation with our first point of discussion, which is centered around the question, what considerations should assay developers take into account when starting the journey from early development to commercialization? Michaela, perhaps if you could speak to this one first from a regulatory point of view.
Yeah, hi, thanks, Cameron, and thanks for having me on today. So I think that's an interesting question because I think a lot of developers that are first starting out on the journey towards maybe creating a new assay or a new diagnostic product maybe aren't thinking about what the ramifications of the regulatory requirements will be later on. And it's sort of my job at TE to sort of remind developers of what those requirements are later on so that you as a sponsor aren't later paying the piper and trying to play catch up to be in alignment with what the FDA or what you might need for a notified body if you're going after a CE mark. So one of the things I like to remind developers about to think early on, it's a common strategy, I think, for assay developers to want to create something and maybe go to market as a research use only product first, and then later think about adapting that to a diagnostic need. And while I think that's a very reasonable approach, there are still, even if you want to go to market initially as an RUO, aspects of the later regulatory requirements you should still think about in the beginning. So for example, if your long-term strategy is to eventually become a diagnostic that, for example, would be capable of gaining a CLIA waiver in the United States, you want to make sure that you designed that RUO product and had product requirements that meet the CLIA waiver like the CLIA waiver designation. So things like simple and easy to use, no reagent addition steps. I think developers need to think about incorporating those things in their RUO product. Again, if they eventually longer term want to take that to a regulated market. Other things I would strongly suggest would be implement design control, even if you're considering starting as an RUO and then maybe becoming a diagnostic later. At least doing the bare minimum of design control is really important. And for those folks on the call or on the webinar maybe don't know what design control is, it's part of ISO 13485. It's a requirement for the FDA. It's a phase-gated development process that requires a lot of documentation, and it may seem overly bureaucratic, but if you do that documentation early, it really saves you a lot of heartache at the end. And I personally think it's important to have, as part of that design control process, really good requirements, product requirements. So I always sort of harp on that at the beginning. But that's sort of my take on it from the regulatory standpoint.
Yeah, and if I could continue on from what Michaela is saying, is the requirements from an engineering perspective as well are very, very important to define early on. They help us to understand the scope that you're trying to meet with your assay, maybe the different indications you like to hit with your specific product, if it's singular or if you're looking to develop more of a platform. They also help us to understand the scope of testing that you're going to need to do later on for your V&V process. So defining those early on is very, very important. Defining the risk profile as well, so understanding the amount of risks you have from a product standpoint, from a user standpoint, is very beneficial to look at early on, as opposed to retroactively doing risk management is not advisable. So absolutely agree with what Michelle is saying on that.
So if I'll jump in here from a sort of manufacturing perspective, for me, one of the really important things that needs to be considered early on is how you visualize the final assay of the format as it's going to be as it's supplied to the customer. So if you're speaking, are you going to say a molecular diagnostic? Is it going to be supplied as a bulk master mix? Are you going to supply it as individual replicates in tubes or in plates? All of these have a sort of big impact on the manufacturing processes and the potential for automation. And as well, further on from that, how big a throughput you can do and how big a batch size you're able to prepare at once. So if you have a clear idea of your product at the start of development, what it's going to be, that can sort of really help you make your manufacturing processes as efficient as possible and smooth that transition into manufacturing there.
Yeah, just to add, a really interesting topic, because often people tend to think quality regulatory can be a last task in the line of development and getting approval, but one certainly needs to consider it early on because that sets a framework of how you develop, how you design, how you organize your activities. For example, reagent QC. The design control allows you to identify risks at every components, every process and steps. And then how you mitigate those risks is, okay, do I need a QC assay for this particular reagent? Then you identify that, and that helps. So design control certainly is a good framework to start with. And then certainly, Michael's point, thinking of how FDA will look at your assay, what are the studies needed, and so, for example, cross-reactivity, right? So, or interference studies or stability studies. Certainly, having this conversation early on with FDA sometime might help setting your work, and so that you are not at the end saying, okay, actually, this is not as stable as we want it to be. And so having those tasks identified and resourced early on.
So sort of coming full circle on that point too, and Brian alluded to it as well, thinking about the studies that you would need for design verification or design validation, going to the FDA early, if the FDA is the regulatory body you're interested in, doing a pre-sub early, even if you're envisioning you're not going to market with a diagnostic for years, it's never too early to engage the agency in a discussion.
No, brilliant, thank you. And Fraser, you touched on formulations there, which sort of leads us nicely into our next point of discussion. What steps can be taken to ensure that the chemistry or formulations is robust enough to avoid, those issues that could turn up further down the line?
So, I mean, if you're looking at the issues that turn up further down the line, the biggest one that often comes up is component stability. Often in sort of development, stability is only really sort of given proper consideration at the end as people are starting to collect data for submission as part of the regulatory process there. So at this stage, you're already too late. If you do identify any problems, you're going to have to go right back to the formulation and sort of repeat a lot of the validation you've worked using your modified assay. I think the most significant step that you can sort of take to this is continuously throughout the process conduct small-scale accelerated stability studies, particularly as you're sort of trialing and adopting new formulations. I mean, to me, the evaluation of assay stability, it should be seen just as important a part of the development process as it is evaluating the assay performance. And sometimes it's too easy to just focus on the assay performance itself. Another thing, which I think particularly with respect to point of care assay testing, is the cold chain storage or the availability of fridges and freezers at the point of testing is not necessarily going to be there. So you have to consider how you can protect against that. So one of the best ways for that is to look at lyophilizing the components of the assay itself. Again, this feeds into the formulation aspect of it, because you need to add things like sugars and polymers, excipients into the formulation to help, to give it that stability there of the process. And if you're looking at a lyophilized format and then the testing of the different different combinations and concentrations of excipients should be a really integral part of the development there. You also need to, if you're looking at lyophilization, you also need to consider the use of particular non-lyocompatible components, so glycerol or high detergent concentrations. So this is, again, it's a decision that needs to be made sort of quite early in the process, because you don't want to be getting right towards the end of development and having to go back and revalidate everything there. So if lyo is to be performed in a molecular perspective, one of the best things to do is consider the use of a glycerol-free enzyme preparation right from the start. Although there are sort of methods of removing some of the glycerol from the removable, this does require additional processing, which adds increased variability into your manufacturing processes. So I think back to circle back, I think one of the key things is really testing the stability of each of your components throughout, rather than just as an afterthought at the end of development.
Yeah, and really, as more and more point of care testing becomes the vogue, and it's like for many of those settings, there isn't cold storage at the point of testing, point of need, point of care, and so lyophilization is important. And just I'm re-emphasizing probably is that formulation development has to consider that early on, because a lyophilized formulation may not work as well as a wet formulation, and you need to iterate and develop that as you are testing. So having that early on is really important, because formulation will change, and also that has to integrate in the cartridge. So where you place what format of lyophilized will be compatible with your particular microfluidic device. So that has to be considered too. So.
And if I could just add on formulation from a regulatory perspective, I think one of the questions we get quite frequently from clients and sponsors is, what happens if I need to change my formulation, but I'm already on the market as a diagnostic? Like, what's the impact to that? Do I have to go back and revalidate? Do I have to go back and submit to the FDA? And I think it's important, if you're contemplating, for example, a change from a wet to a lyophilized reagent, or a change in enzyme, or a change in primer, engage your regulatory consultant. We can walk you through the risk-based decision tree so that you can decide whether that would require a whole new submission, or you might be eligible for something we call the special 510K. So we can sort of help guide, what the decisions would be in terms of changing a formulation, or what the impact of that would be.
No, brilliant. Brian, if there's nothing to add, sort of moving on to the next stage, which is obviously when you're selecting a cartridge. So I don't know if you'd like to kick us off talking about how the risk associated with cartridge material selection and design can be mitigated.
Yeah, that definitely dovetails right into that aspect of it. So understanding that the way we approach cartridge development from the engineering point of view is to come from a framework that the assay and its requirements drives the design of the cartridge, as opposed to the opposite. So when we're designing a cartridge, we need to know as soon as possible and to as high of an amount of detail as possible the functions that it needs to perform and what functions are critical for the assay to perform effectively. So we're looking at really early on mapping out the assay workflow, the individual steps, the requirements for those steps, like temperature, time to perform certain steps, or whatever is critical to maintaining that assay functionality. There's a lot of interactions that are important to think of early on. There's chemical interactions when we start to design in a cartridge format that's different than your bench format. There's some chemical interactions, there's some mechanical interactions. And some of those chemical interactions include things like PCR inhibitors that wouldn't normally be there in the tube format that you might perform your assay in, but in the cartridge format where we might need to include things like lubricants to help with moving sliding components that are there for different functions. There's different chemicals, there's different materials that might have to be used for the cartridge. that could inhibit PCR, that could inhibit parts of your assay. There's also material properties that could bind molecules that are critical to your assay, where you wouldn't want that to happen. So we need to understand those assay functions early on to understand if we need to maybe coat the materials used, or use a different material property to either bind or not to bind. There's also mechanical interactions that we want to think about. This has always been very, very important to understand things like heat required for your assay, to understand things like pressure or different forces. So one of the things that comes up a lot in these assays is the thermal ramp rate for PCR or any thermocycling assay steps. So we need to understand, really, is time to result very critical for your assay? And that could lead us down saying we need to have very fast thermal ramp rates. That works really well in a tube, but when you move to a cartridge format, you need to start heating it up on the outside of the cartridge a lot more than on the inside of the cartridge. And with certain materials like a polypropylene resin, for instance, that you would normally use in your bench top, your labware, that has a lower softening point, so it can usually survive up to about 100 Celsius before the material starts to get soft, and that impacts the structure of the cartridge. So we might want to choose something different that can handle those temperatures, like a polycarbonate or a COC or a host of different resins. But we need to understand what is critical to your assay so we can choose the right resins. There's also things like pressures. If we need if the assay requires higher pressures, the cartridge design will need to accommodate for that, and we just need to make sure that these things don't play off each other in a negative way. We're also looking at things like optical clarity. If you're looking at fluorescent or other optical detection methods, we might need to choose, or we would need to choose, a resin that doesn't absorb any of the wavelengths of light that you're interested in. So that will really drive us down a resin choice and down a design choice to accommodate that. And a lot of these things, in an individual component level, they work over here. But when you start to integrate all of these different functions into a cartridge, they often can play off of each other, and you need to take those things into account because there may be design trade-offs. So when we've been doing this a lot, for us to be as most effective as possible from an engineering standpoint, we want to look early on. That's been kind of the theme of this, is early on we need to look at if the assay is compatible with the resin choices. And so one thing we can do is we can give you the pellets of the resin, or small chunks. So we can do something really simple, like sending you the pellets. You can put those in your actual, the tubes that you perform your assay on. You sort of soak the chemicals in those resins for as long as it would be experiencing that in the cartridge. And then you perform the assay, and then you can remove those. But you can do that. We can send a bunch of different chips of different types of resins, and you can do that assay compatibility testing. We can also go to a more complex route, which is making what we call coupons, which are just small subsets of the actual assay function in a cartridge format. So we can start to isolate the thermocycling region, or a capture region, or a washing region, and we can understand individually. So removing other outside variables, we can understand how the assay would function with different material choices, or different geometry choices, or different interactions. But understanding those things as early as possible helps us to map out the assay function, the critical parameters, and understand the tolerances of temperature ranges, or force ranges, or pressure ranges that the assay can survive.
And yeah, thank you, Brian. And has anyone else sort of had similar issues or experiences that they'd like to share?
Yeah, we're developing a POC device, and that had assay the cartridge and the instrument. And to Brian's point, exactly, when you transport a tabletop assay into a cartridge, into an instrument, the issues you face are the same. We face the same issue, like just heat it to 72 degree inside the cartridge, the fluid, you may have to go and overshoot and then come down to 72 to do it in time. So understanding the thermal profile, the thermal absorption is important. One other thing you mentioned, Brian, is the PCR inhibitor. Yeah, we used to test different resin beads in the assays. If it leaches out, something that inhibits reaction. Another thing we experienced is, which is not often appreciated, is absorption by the cartridge material of enzyme. So if you have a microfluidic device and you're moving stuff from one chamber to the other, during the transit, enzymes are very low concentration and they can get absorbed on the wall of the microfluidic device. And so you have to work with all the issues that cartridge may pose, and understanding that early on is including, as you're moving stuff, it creates bubble, right? So that can clog the channel or can create other optical issues. So understanding all that, and then so having a prototype and working with the cartridge manufacturer early on, what they have seen in the past, it really helps. It really helps.
No, thank you. And Fraser, is there anything?
I was going to jump in from if the lyophilization, if you're going to be using lyophilized reagents within the cartridge, again, that needs to be considered. So is it an option to lyophilize directly in the cartridge? Because again, that will need to be taken into cartridge design, because as well as reaching high temperature, you have to reach low temperatures. So you might need a wide temperature profile there. If you are looking at lyophilizing directly in the cartridge, there's other things that need to be considered. So the critical drying parameters are going to be affected for your formulation there, because the product's going to be in direct contact with the shelf as well. You're speaking potentially reduced volumes, because there's a lot. Each cartridge will be taking up additional space that a very small amount of reagent isn't in the freeze dryer. So the alternative in this case is to look at using something like lyophilized beads, so very tiny droplets of freeze-dried reagents, which allows for much larger batch sizes. Again, they also come in with their own problems their own difficulties afterwards, because I think Dipankar touched on this earlier, but you're going to have to actually place those beads within the correct location of the cartridge as well. So there's a lot of considerations, even from a purely lyophilization perspective, that will need to be taken into account there for cartridge design. Yeah.
And something, Fraser, you talked about in the previous question, but storage of these materials on a cartridge format is very it can be pretty tricky, because there are trade-offs between temperature. You may want to lyophilize. Lyophilization is often the best solution for some of these more sensitive enzymes, but you need to take into account, this will come into play on how you store. If there's liquid reagents, like some just water or salt buffers that can be stored, and if they're stored next to more sensitive reagents, it just makes the design a little bit more complicated than you had initially considered. If it does need to be frozen, then that'll impact how the design is stored how the cartridge is designed as well, because some resins may not handle freezing and thawing. And if I could, one more thing that was also mentioned is just taking into account that assay workflow. When it's read out loud, and we may notice there's a step that was called out that was hand pipette or mix by hand or shake or something like that. Understanding those physical interactions of the assay, but there are physical interactions that are beyond the chemical interactions, and those things, like if there's a quick centrifuge step, those things need to be mimicked, or they need to be created on a cartridge. So understanding those more from up front, they look like minute details, but when you actually read them out loud and understand those need to be replicated on the cartridge, it's really, really important to know that up front so we can design it correctly. Yeah.
No, thank you all for your insights. Oh, Dipankar.
I was thinking also of the regulatory side of it, right? Often developers will run a lot of early studies in 96-well plate or centrifuge, and then when they transition, they often assume some of those holds true. So they may complement the submission with some actual cartridge data and some non-cartridge data. So from a regulatory point of view, what do you think, Michael, that how it is handled?
Yeah, so if we're talking about the FDA, for example, the FDA is interested in clearing your device's system. So all of your data, whether it's design verification or design or clinical data validation data, has to be on a production equivalent system. So anything you may have done when the assay or prior to the assay being ported onto the cartridge wouldn't be applicable for checking those boxes with the agency. It really needs to be demonstrated in the production equivalent system.
No, thank you all. Sort of leading into the next question, the next point of discussion, why is sort of supply chain management and the strategy to address that so valuable?
Yeah, a really interesting point. And the way supply chain management is defined in this space is includes the development, includes procurement, qualification of those reagents, managing inventory, getting it to the customer, and at the end, disposing the used test. So that entire thing comes under supply chain management. The part that becomes very critical early on is if you have key raw materials, right? You have to make sure that you are engaging the right partner who will provide you quality ingredients, consistent lot-to-lot material, because that is as important as the quality. And then they do have a robust manufacturing facility and infrastructure to support you. So all of us saw what happened during the COVID days. Supply chain became the critical bottleneck. The demand for testing was far outpacing the supply of testing material. So enzymes, oligos, purification beads, all of those became limiting. And so having the right partner in this space who can not only supply the quality and the consistency of reagents, whether it's oligo, whether it's enzyme, buffers, or beads, it's also important to see their resilience against market fluctuation, right? And so how do they manage their inventory? How do they manage their production? And do they have backup? Do they have buffer? So that's why it's important early on to thinking about not just the assay. It may have worked with certain suppliers, certain vendors, raw components, but is that the right partner in the long run? After you have gone to market, will they still be able to support you the increased demand, and will they be robust enough to manage the fluctuation in the supply chain? And many suppliers, they may have manufacturing in-house, but many even secure some of the reagents from another country, maybe another supplier, right? So understanding that is important. So auditing your supplier is important. And one of the things we have seen is that there is lot-to-lot variation, whether it is enzyme, whether it is oligo, probe, dyes, etc. So understanding the robustness of the assay to those variation. What's the gut banding that you have done in terms of tolerance of the assay to fluctuation in enzyme quality, concentration, etc. And then are those reflected in the way you're QCing your incoming material, the way the supplier controls the release of those material? So understanding all of it is really important, because you may say, yeah, it works fine, tested one lot from one supplier, but then when you go to actually practice, you may be surprised that the next lot does not get the same way. So I think it is really important to work with a robust supplier, whether it's oligo, enzyme, or beads, or even plastic components, right, resin components. And lyophilization partners. So I think all of it has to be thought through in the beginning, the go-to-market strategy, not from just commercial point of view, but also manufacturing scale-up point of view, and do we have the robustness in keeping up the demand if it goes up.
A little bit to your point, it's I haven't ever seen it been this difficult for several years, but getting hold of plastic resins has been still just weirdly difficult. So yeah, understand that that's going to be a force at play for, I don't know how long, but yeah, there's been force majeure of certain resins that are critical for things out of our control. But yeah, the supply chain is still fairly difficult for some certain elements.
And to chime in on some of your comments about quality and lot-to-lot variability. So, I mean, it's an incredibly important part of ISO 13485 and your quality management system that you're evaluating these vendors and putting in, audits, having some quality agreements with them. Especially if you know the tolerance that you need for a particular material, having that contractually sort of locked in is key. And then when you go to do your validation studies, at least with the FDA, there's an expectation that you're including multiple lots of key critical components in your assay, in the lots that you're using for your validation. So whether that's multiple lots of enzyme or multiple lots of primer, all this sort of comes out of your risk determination during the design, control, and development phases. But yeah, you've got to flex all of those lots in what you're evaluating.
No, thank you. I think sort of the next point of discussion, and it's popped up a few times, sort of about point of care and at home testing, and indeed in the field. What sort of additional considerations, and I know we've touched on it before, sort of come when you're approaching it from that sort of, that's the end point? Is there anything else that sort of is important to consider?
I think to me, go ahead, Fraser.
I was just going to throw a couple of very basic questions out there that are going to be important in terms of who's going to be performing the test and what sort of level of training is going to be required to do this. Ideally, you want, if it's not necessarily highly trained individuals, you're going to want the results to be very easy to interpret. So something that gives a simple sort of yes or no, positive, negative readout. And then as well, so the sample matrix that's going to be tested, how easy that is to collect, because again, that will potentially require additional training there. So these are just very basic questions, but they feed in right at throughout the development there.
Yeah, during the COVID, of course, point of care testing got heightened interest, and everybody wants to do it so that it's a democratization of the test in a sense, and it's a much more rapid too. So from that point of view, the test actually surprisingly maybe somewhat not common sense. It has to be more robust than a lab test because the users are not trained, and they will challenge it in as many ways as you can think of, right? And so if you in the lab, you could easily pipette 20 microliter and add it to there. But now if you're doing 2 drops can be very varied, and somebody might even put 3 drops, right? So you have to test the robustness of the assay and the workflow way more than if it is just a lab test.
And Michele, you brought up several times that the risk profile early on, or risk management, but as you look into that, if it's critical for the assay to interpret the result correctly, as has been said, there may need to be different assay controls that are put in place to ensure that there are no false negatives or false positives that the user can't find in such a way that it could harm the user, or the result could harm the user. So having to understand that risk profile early on is very, very important for the design of the assay to ensure that it's something that can be verified and something that the FDA will be okay with.
No, I think that's an important point, Brian. I mean, the pandemic sort of demonstrated to everybody that we are capable of running these tests at home and interpreting them, but the agency did put some requirements in place, like you shall have internal controls. And even with like the monkeypox outbreak, there's been requirements that your test has to have multiple targets. So you can't just detect one target and be diagnostic. You actually have to detect a couple. But I think with the move towards, more and more home testing, this door's kind of been opened because of the pandemic to more and more testing at home. I think there'll be an increase in the number of tests that will be read using cell phone apps and sort of taking the subjectivity out of it with the lay user at home interpreting it. So that's sort of a new emerging territory for the regulatory side too.
No, thank you so much. I'm afraid we must move on time-wise towards the Q&A session. So thank you all for your really considerate answers. We hope that everyone listening has enjoyed the discussion so far. We will now be moving on to the last part of today's webinar, the question and answer session. Of course, please feel free to continue asking questions using the tab at the left of your screen. Simply hover over that purple speech bubble icon and click to ask your question. Brian, it's great to have you back on the line. I believe we lost your camera for just a moment there. So to kick off the Q&A, we have our first question. And that asks, how do you think assay development will evolve in the future? And is there a resistance to adopting new innovations? I don't know who would like to go first to address that one.
Yeah, I can go first. I think it's a really interesting question. I think, as many of you mentioned, point of care testing in the earlier section, isothermal amplification is one area that has gotten a lot of attention in point of care testing. In terms of newer technologies or innovation, in the last few years, CRISPR-DX has come into market. So that's using the CRISPR technology and which is also isothermal. So I think the combination of CRISPR with other detection technologies is really a promising area. There are already few that have at least made it to the field testing, and I think that holds a lot of promise in future. There are a few other isothermal technologies being tried. So those involves a modification of LAMP or other isothermal technology. So I think those will be something to watch out for, because in the end, if we are going to take diagnostic to the customer, isothermal is a much better option than cycling of temperature.
I mean, for me, I guess what we've seen over the last couple of years is, especially has been demonstrated to the wider public, is just the benefits and potential of molecular diagnostics in particular, and just how they can help with quicker diagnosis and quicker actions. And sort of part of that's been due to sort of the big proliferation and sort of the number of different point of care testing platforms available. Most of those, they've used COVID tests to demonstrate the equipment, validate it, show how good it is. So there's been a big increasing number of more and more of these on the market. So I think now with the COVID testing beginning to reduce a little, what we're beginning to see is the platforms diversify and they sort of go into the areas in which they were particularly designed for. But personally, what I think I'm going to see in the future is a handful of particular platforms are going to begin to dominate the market, and we'll see that most new assays are being developed to work on specific platforms, and that's certainly the way I see things going myself personally.
Great, thank you. And Brian, Michelle, is there anything you'd like to add? Michelle,
yeah,
go on, Brian. Yeah.
Oh, sorry, probably a delay. Yeah, from an engineering perspective, we've seen a lot of people looking at personalized at home, like we've mentioned before, kits or test kits that can be very easily used by someone at home, but are very sensitive and provide meaningful, quick and meaningful results that can be actionable by someone at home. Yeah, there's not an enormous amount to add, but a lot of the assays are being developed with that thought in mind, that people at home can take the action into their own hands for their own health.
And I don't have much to add on the topic, other than, like it's been said before, I think the pandemic has demonstrated that users are able to run these tests at home, and we'll sort of start to see a surge in approvals or at least submissions for more and more, like a broadened menu of at-home tests. I don't think that there's anything specific to the type of technology that would be limiting to that. I mean, obviously technologies that the FDA or the regulatory agency are more familiar with may have a slightly lower burden. But that's not to discourage or say that new and novel technologies, like for example, the CRISPR, couldn't potentially go to an at-home pathway. It really just depends, again, like Brian said, how easy it is to use and interpret the result. So.
Brilliant, thank you. And Michelle, sort of to that question, do you think the adoption of new technologies, people are worried to sort of take that first step in terms of IVD approval?
I'm sorry, Cameron, I didn't understand the question. Can you repeat?
Yeah, sorry. So in terms of sort of adopting some new technologies as they come out, do you think there's that worry for some manufacturers to sort of take that first step in terms of IVD approval?
Oh, yeah. So I think sometimes there's a lot of apprehension in potentially addressing or approaching the agency with something that's very novel. There's a lot of sometimes of doing panels that are Me Too's, right? It's certainly not an insurmountable hurdle. I mean, there are we've talked about the need to do pre-submissions and discuss with the agency, but there's other mechanisms that you can use to sort of engage with the agency early if that's really a concern of yours. Novel technologies are ideal for something like a breakthrough designation, which could be really appealing if you're a startup company or you're looking for some investment. So regulatory can help with evaluating the potential of your device for that kind of designation as well.
Brilliant. No, thank you. We've had another question come through asking what the critical assay parameters are that needed to be understood when designing a product.
I can start the discussion probably. I think, yes, there are a lot of things as we discussed early part of the webinar. Reagents is one thing, but also the parameters within which the assay will work, whether it is time, temperature, optical signal, the channels, etc. I think understanding the whole workflow and identifying the boundaries for each step is really important, and that's where something like guard banding study becomes important, knowing where it will fail the assay and testing the failure points. Yeah.
No, that's a really good point. The guard banding studies are very critical for, especially even as you're going into the designed and the manufactured part. There will be manufacturing tolerances of the plastic components of the full assembly. So needing to understand not just what is the nominal or the middle of the road design. For instance, if something's meant to work for exactly 65 degrees Celsius, in reality there is no exact. There's an average, and then there's a tolerance to which you're going to be wobbling above and below. And there's tolerances for every component of the assembly. So understanding the ranges of the requirements. So is it meant to work exactly at 65? Can it work and still meet your efficiency and sensitivity requirements down at 62, up at 68? Understanding those ranges. Additionally, for the plastics, because the assay will need to be held within plastics, can it work within resins that are readily available? So can it work within a polycarbonate resin? Can it work within? Does it need to work within a polypropylene resin that's treated to some degree? So there should be a good amount of testing done early on with the resin choices that we would want to use to understand will this impact your assay.
Great, thank you both. The next question I have for you all, and perhaps Fraser, sort of what you touched on. How do I know whether my assay is better suited for air drying or lyophilization?
Yes, a very good question, that, and sort of one that comes up quite regularly. for us. But it's really a horses for courses type of approach, and there's no a definitive method for each. I mean, the differences, I guess, for people who are less familiar. So lyophilization, you involve freezing the product and applying a vacuum to remove water from the product and via the process of sublimation, whereas air drying would generally involve heating the product to remove the water via evaporation. So as a general rule, air drying does remove less water, and as a result of this, the products can be potentially less stable. As well as that, if you're dealing with temperature-sensitive components, particularly something like enzymes, they might not be as functional after being heated in this manner as well. So in that case, lyophilization might be a better approach. Whereas if you're dealing with more sort of stable components, let's say nucleic acids or oligos, those tend to work quite well with an air drying approach. In general, though, really have to consider it on an assay by assay basis. But what you find is air drying will generally be quicker and a cheaper option there. But it really depends upon the key components of the assay or the reagent itself.
If I could add on really quick to what Fraser's saying, that there will need to be some consideration for the assembly steps. So for some assays, depending on where the reagent needs to go, that it may be necessary to dry it down onto a surface. If you can't physically put a bead into an area and then seal it. So those are one of the things that'll need to be considered during the design. It may be driven into one design modality or another.
Of course, yeah.
Great, thank you both. Michelle, the next question sort of focused on approval. So the audience member asks, have regulatory bodies like the FDA changed their testing approval for their testing and their clearance process in the face of the pandemic? And would you say the sort of process is more responsive now as a result?
Yeah, I would say like the regulatory pathways and the processes haven't themselves changed. Now, obviously, during the COVID-19 pandemic, we had access to the emergency use authorization pathway, but unless a sponsor has government funding or, are adding like significant value in an amendment to an existing EUA, that door is pretty much closed. I will say during the height of the pandemic, because so much was coming in, to the agency for review, there was a long period of time where they weren't reviewing submissions for anything that wasn't COVID-19, and that period has now passed too. So we're seeing the agency get sort of back to meeting its target MDUFA timelines for review of pre-subs. They're reviewing pre-subs that aren't related to COVID-19 anymore. So it's sort of a return to normal. Now, that being said, even though the processes are the same, and I think we've said it in this seminar several times, I think you will see moving forward with the agency is potentially more at-home tests coming in for approvals and a broadening of the menu of tests that might in the future become available over the counter.
No, brilliant, thank you. So for another question, with technologies like LAMP, sort of loop-mediated amplification, sort of coming into that point of care and sort of point of need standpoint, sort of that untethering from thermal cyclism and moving to an isothermal standpoint, do you think you'll see more technologies sort of lean towards that as those processes are developed, and how do you think that will sort of affect the design process going forward?
I can comment on, I've been associated with LAMP technology for a while and the enzymes. Yes, I think really LAMP was probably one of the most popular isothermal amplification technology. And there are a few marketed tests, IVD tests, that utilizes LAMP. I think the field may see a increased use of LAMP per se because the patent held by ICANN is about to expire. So that might allow more startups or companies to use it. So that's one area. And then there are a few companies that are iterating the lamp per se, but still isothermal. And I think those we are going to see as well. Yeah.
From a design point of view, it certainly it makes the thermal control a lot more simple because you're not trying to rapidly and accurately increase and decrease the temperatures. But yeah, it's definitely preferable from that point of view.
And again, cutting back to some of the other things that we've discussed earlier on in the conversation as well, some lamp results can be very easy to interpret for a lay user as well. There's less complications involved there than there are for some of the other more traditional alternative qPCR based alternatives. They're just much easier to interpret. Yeah.
Brilliant, thank you. Well, I think we've got time for one more question today, and that's sort of a follow-up for you, Michele, and obviously more broadly, is sort of when should IVD developers engage with reagent suppliers sort of in the development process?
So I would suggest that the engagement with the suppliers, I mean, from a quality perspective, you have to have all of those supplier contracts in place early on. Especially with the supply chain issues we've already discussed.
If I can add to that, other benefit of engaging the supplier early on will be that they may be able to help you customize. Like if you don't want glycerol in your enzyme, if you want a different detergent in your enzyme storage buffer, or a different concentration of the enzyme. So those customization are doable. So engaging the supplier early on and talking through all the options, what they can do, and they may have even a different enzyme which does the same thing. So it might help having that conversation.
It's also always great to understand early on if you're going to meet your cost of goods target, or if the reagents they're choosing, like you said, are readily available. If you could suggest something that's not novel, it's great for you to understand what your suppliers can and can't do before you start forcing them to do something.
Brilliant. Well, I'm afraid that is all we have time for today. I'd like to thank our expert speakers again for today's discussion and, of course, the Q&A session. And a big thank you to everyone joining us online for submitting your questions. We hope that you found this a really worthwhile session.